Good morning, everyone. Thank you for the introduction, Axel. Today, I'm going to be giving a brief overview of my research regarding the evolution of language within the hominin lineage, with specific focus on the evolution of Wernicke's brain area. This research was conducted as an undergraduate and was published in Elsevier's Progress in Brain Research series. I'm going to cite this paper at the end of the presentation if anyone's interested in following this topic up. So before becoming too niche, I'm going to give some background on preceding studies of language evolution. The suite of morphological, genetic, and behavioral traits needed for human language did not just magically appear in Homo sapiens as a polished final product. Rather, they arose piecemeal in different species following the divergence of the hominin and pan lineages about 7 to 10 million years ago. Human language is also not an evolutionary gold standard. We cannot discount the possibility of different and valuable forms of communication in non-human hominids. Studies of language evolution often use chimpanzees as a proxy for the last common ancestor of the hominin and pan lineages, describing language-related traits in hominins as closer to the ancestral form if they are chimp-like. This is an imperfect approach, as of course chimps have been evolving independently for as long as humans. Yet, in the absence of this last common ancestor, chimps are our best available stand-in. So keeping these things in mind, we can ask the question, which, if any, of the species within our beautiful ancestral tree were speaking? As listed here, several language-related traits have been analyzed within the hominin lineage. As audition is essential for processing languages, researchers have examined hominin inner and outer ear morphology and modeled sound power transmission. The size and shape of the hyoid bone dictate range of movement for the tongue and larynx. Unlike humans, chimps have this cup-like extension to their hyoids called a bulla, which has also been noted in Australopithecus afarensis. With regard to the vocal tract, the 1-to-1 ratio in humans between oral cavity length and pharynx height, which enables vowel production and which is absent in chimps, has also been recorded in Homo heidelbergensis and Neanderthals. The area of the hypoglossal canal, which transmits the nerve allowing fine control of tongue muscles, was proposed as a means for distinguishing the vocal capacities of modern humans, but no significant difference in canal or nerve area was found between humans and chimps. Other researchers have examined a different canal, the thoracic vertebral canal, enlargement of which in humans relative to chimps may be related to breath control during speech. This canal is within the chimp range of variation in Australopithecus and Homo ergaster. Material culture may also intimate the language abilities of non-human hominids. For example, experimental research suggests verbal teaching, as opposed to teaching via emulation or imitation, may have been a prerequisite for the transition from mode 1 Aldoan to mode 2 Acheulean lithic toe production. And finally, though it is of course an oversimplification to draw a direct line between single genetic variants and language abilities, the gene FOXP2, which differs by 2 amino acid substitutions between humans and chimps, and has been found to be human-like in Neanderthals and Denisovans, can cause developmental verbal dyspraxia if disrupted. Now I'll move away from the non-neural lines of evidence for language evolution into my area of interest, the neural evidence. The human brain has 2 main language areas. Broca's area, highlighted in yellow, lies in the posterior inferior frontal gyrus. On the human brain surface, its borders are roughly demarcated by the inferior frontal sulcus, the inferior precentral sulcus, the sylvian fissure, and the ramus horizontalis of the sylvian fissure. Just a quick note here, when I refer to sulci and gyri, I'm referring respectively to the furrows and ridges on the brain surface. Damage to Broca's area can cause non-fluent aphasia, whereby an individual's speech is severely reduced and their sound formation is clumsy, so it's been connected with language production. Conversely, damage to Wernicke's area can cause fluent aphasia, or difficulty comprehending speech. As highlighted here in blue, in humans, Wernicke's area sits in the posterior superior temporal gyrus, near the caudal end of the sylvian fissure. Homologs for Broca's and Wernicke's areas have been identified in chimpanzees. For Wernicke's area, comparisons between relative size, asymmetries, and sulcal organization between humans and chimps have not been adequately conducted. This study aimed to start to plug this research gap and apply resultant findings to fossil hominins, determining whether their Wernicke's areas more resembled those of chimps or those of humans. As the brain itself does not fossilize, how then do we study these brain areas within the fossil record? The answer is endocasts, or casts of the endocranial surface. Endocasts can be naturally formed via sediment accumulating in the cranial vault during fossilization, as in the case of the Tong child specimen pictured here. Synthetic, so made by pouring molding agents into the cranial vault. Or virtual, made by creating 3D models from X-ray or CT scans, as was done for the Littlefoot specimen. Endocasts give us information about the brain's shape, size, vascularization, and sulcal organization. Unfortunately, fossil crania are frequently incomplete or distorted, so identifying sulci and gyri is tricky. There is also some debate as to the validity of assessing brain organization via imprints on endocasts. Endocasts don't perfectly reflect the brain surface, and sulci don't perfectly mark functional borders of brain areas. Yet, as goes the common adage in paleontology, we have no other available proxy for studying ancient hominin brains. Thus, endocast studies continue, along with attempts to augment them and cognizance of their limitations. So my particular study, as stated, was a macroscopic and qualitative analysis of Wernicke's area of the brain. My sample consisted of 5 chimp brains, 4 human brains, 1 Paranthropus robustus endocast, and 3 Australopithecus africanus or Prometheus endocasts. Sulcal designations were made by consulting brain atlases, such as that of Conley. Chimp and human brains were visualized using 3D Slicer, and hominin endocasts using ParaView. And after examining visualizations, screenshots were taken for each hemisphere, upon which sulci in the vicinity of Wernicke's area were marked in specific colors using the draw function of Microsoft Word. So what were the results? Firstly, in chimpanzees, a shorter Sylvian fissure and/or caudal superior temporal gyral bulging were observed in the left hemispheres of some individuals, which may indicate leftover right Wernicke's area size dominance, as reported by other researchers. In humans, overlap between the middle and superior temporal sulci in both hemispheres, which was not noted in any chimp hemispheres, may signal a relatively larger Wernicke's area in humans than in chimps. Furthermore, fragmentation of the superior temporal sulcus exclusively in human left hemispheres may be a product of leftover right Wernicke's area asymmetry in humans, as also found by other researchers. For the endocasts, only 1 hemisphere was available per individual, so analysis of interhemispheric asymmetries weren't possible. Overlap between the middle and superior temporal sulci was documented in Paranthropus, but not in any of the Australopithecus specimens. Meanwhile, caudal superior temporal gyral bulging was observed in all Australopithecus specimens, but not in Paranthropus. This was taken to very, very tentatively indicate a more human-like Wernicke's area in Paranthropus and a more chimp-like Wernicke's area in Australopithecus. This study had considerable limitations. First and foremost, subjectivity of sulcal ID. In human brains, temporoparietal convolution caused marked sulcal fragmentation and overlap, so different observers may have given very different designations to my own. Inter or intra-observer error was also not determined. A possible solution to this might be the approach developed by Labra et al., which combines dice scores and mean-minimum Euclidean distances to quantify variability between sulcal demarcations. Technologies for automatic sulcal ID are also being developed, which will hopefully reduce subjectivity. The second issue lay in small sample size. For humans and chimps, this was due to time constraints, so could easily be inflated. The hominin endocast sample is inherently limited by preservation, but may be increased by current efforts using AI to reconstruct and detect endocranial markings on highly fragmented cranial vaults. Use of endocasts for hominins and 3D brain models for humans and chimps may have created inconsistency in my observations. I perhaps should have also generated virtual endocasts for the humans and chimps. Another problem is that the precise functional borders of Wernicke's area remain unclear, which adds compound error to my analyses. Functional or fMRI studies in humans and chimps may help resolve this. It is also difficult to extrapolate any correlation between these findings and actual language abilities. In future, we need to investigate the impact on language abilities in humans of having a larger, smaller, or asymmetric Wernicke's area. A final issue is that though I conducted my analysis in 3D models, results were presented in a 2D format on which surface bulges were less evident. A better solution may have been to color map the relief of the brain or endocast surfaces prior to screenshotting.
Harmony, you have 1 minute left.
Thank you. This research laid the groundwork for my paleoneurological interest, which is translated into my current PhD thesis. My focus has shifted slightly from brain organization to brain vascularization, and I'm seeking to track metabolic accommodation of brain changes in the hominin lineage using osteological proxies. As well as investigating these proxies in surface scans of fossil hominin specimens, as pictured, I'll investigate their intraspecific variability in modern humans, as well as the level of actual variation in brain blood flow and metabolism in living persons. So that's all from me. Thank you for your attention. Any follow-up questions could be directed to my email or LinkedIn, which are on the screen, or to the Discord server.